Hello everyone, welcome to the new Ant Biology webinar series presented to you by the Translational Outcome Research Group of the Department of Zoology, University of Calcutta. This new series aims to celebrate the latest advances in the field of life science along with its relatedness to other disciplines. I am Bijoy and I will be a moderator for today's session. Before we begin, I would like to inform you that you can post your questions in the YouTube chat box and we shall moderate them at the end of this lecture. You are free to interact with each other and with the speaker. If you have any questions or comments meant for the speaker or for us, feel free to email us and we shall address them to the best of our abilities. And you will also send us your feedbacks through the feedback link, which will be provided in the YouTube chat box near the end of the session. Unfortunately, our convener, Professor N.R.I. Banerjee of the Department of Zoology, University of Calcutta, is unable to join us at this moment, though she was looking forward to interact with our speaker. So I would like to take this opportunity to welcome our speaker today, Dr. Oishee Chakraborty. A very warm welcome to you, ma'am. So before I hand over to ma'am, I would like to give a brief introduction of today's speaker. Today we have none other than Dr. Oishee Chakraborty with us as our speaker. Oishee Chakraborty obtained her bachelor's degree in geology from Benares Hindu University. She then joined the Integrated Masters of Science and PhD program at the National Center of Biological Science (TIFR). She received her PhD pursuing studies on the modulation of cellular signaling pathways by high-risk human papillomavirus 16E6 oncogene variants in cervical cancers and continued there as visiting fellow. After 1 year stint as a research fellow at the Department of Pathology, Harvard Medical School, she joined the Cell Biology and Metabolism program, Eunice Kennedy Shriver National Institute of Child Health and Human Development, National Institute of Health, and continued as a research fellow till 2009. After this, she returned to India, became a principal investigator at the Structural Genomic Division, Shah Institute of Nuclear Physics in 2010, and continued there as an associate professor. Her primary research interest is to understand the molecular pathways that lead to extensive neuronal death in the late onset neurodegenerative diseases. So, she has several peer-reviewed research articles, original reviews, and book chapters to her credit. She is recipient of the prestigious Janaki Ammal Women Scientist Award from the Department of Biotechnology. We are glad to host her today and excited to hear from her. Dr. Chakraborty, the stage is all yours.
Thank you, Vijay. So I start, yeah, I will share my screen now and let's get started. Thank you much for your introduction. And so this this talk is primarily aimed at the
sorry for the interruption. Ma'am, sorry for the interruption. Can you please hide the bottom bar that is showing on your screen? That hide option. Can you click on that?
Okay.
Okay. Thank you, ma'am.
Is that better now? Okay.
Yes, ma'am.
Yeah.
All fine.
So this is primarily aimed. This talk is primarily aimed at the research scholars, the postdoctoral fellows, the postgraduate students, and also the undergraduates. So some of my senior colleagues, if there are any in the audience, would find a few things redundant at times, but these have been purposefully added to make this talk as lucid as possible for the students and young students and researchers. So today I will talk to you about something you have not heard, probably most of you have not heard a term, Reticulo-mito-phagy. So there are three things involved in this terminology. One is reticulo, which stems from reticulum or the endoplasmic reticulum, mito or the mitochondrial, and phagy coming from autophagy. So this is basically the turnover of two organelles of the cell simultaneously, the endoplasmic reticulum and the mitochondria. So let's get started with the first terminology, which is the reticulo, or the endoplasmic reticulum. The endoplasmic reticulum is the seat where the secretory and membrane proteins are made. Then they are transported to various locales in the cell or outside. It can be at the membrane as transmembrane proteins or GPI anchored cells of the proteins. They can be into various cellular organellar membranes. It can be the Golgi, the other organelles like the and also secretory proteins. So that this is how the endoplasmic reticulum looks like under a confocal microscope. This is actually expressing a GFP tagged endoplasmic reticulum retained protein. And if you look into the production and the transport of these secretory and membrane proteins, each step is very tightly regulated. Mislocalization leads to diseases, of which there are many such diseases known, like really hundreds and hundreds of such diseases known, and many of them lead to neurodegenerative diseases. A good proportion of them are neurodegenerative diseases. So let's take a step back. Now, what is the purpose of this endoplasmic reticulum? The endoplasmic reticulum, by virtue of the ribosomes which can get associated with it and give rise to the rough endoplasmic reticulum, proteins are made, then they are translocated either completely inside, and these are the ones which get secreted out, or they are embedded at the membrane, or like transmembrane proteins. Here, some modifications, some glycosylations would occur, and the protein would get further translocated to its final location, be it the plasma membrane, be it the endoplasmic reticulum itself, or the Golgi, or even the lysosomal membranes, or various other vesicular membranes. Now, I told you each of these steps are very well regulated. However, if there is a problem in a protein, so suppose there is a problem like the marked in red in one of the secretory proteins or in some of the transmembrane proteins, then these do get recognized inside the endoplasmic reticulum itself, and they get transported out of the endoplasmic reticulum back into the cytosol by a process called retrotranslocation. By virtue of this process, the protein gets ubiquitinated and or modified by the ubiquitin moiety, and finally gets degraded at the proteasomal complex. So this is a very tightly regulated protein quality control mechanism which happens at the endoplasmic reticulum, and for obvious reasons it is called ERAD, or ER associated degradation. So here these misfolded wrongly modified proteins, they retrotranslocate and then finally get degraded. If these are correctly folded, then they go ahead to get exported out by their normal channel of protein transport. The ER is not just the seat of protein production and quality control, it is also the seat of calcium storage, lipid biogenesis, and it is an essential component of the endomembrane system. Now, one more thing which is becoming more and more obvious with time now is that It is also the place where autophagic membranes, it also is the source of autophagic membranes. So what do you mean by that? So when an autophagosome is formed, it is, so this is the how an autophagosome looks like. But what happens is that the ER can actually help in the de novo deposition and synthesis of the isolation membrane, which can curve in and form the phagophore. This can get completely pinched off and form the autophagosome. So the ER is also one of the richest sources of membrane for the formation of the autophagosome. But bear in mind, please bear in mind, this is not the only source. Autophagosomes can originate from the mitochondria, they can originate from Golgi, they can even originate at the lysosome. So autophagosomes can get their membranes from various sources, but the endoplasmic reticulum is one such source. Now, to understand any mechanism, one of the best ways which has been done time immemorial is to perturb it, so that the well-oiled the machinery of the cell, the functioning of this, it stalls, and moment it gets perturbed, we can start understanding how these perturbations are affecting a particular cellular mechanism. So one of these is protein mislocalization. So when a protein gets mislocalized, it kind of acts like a wrench in a gearbox. It causes, so a small wrench can actually stall this majestic gearbox by very small perturbations. It can cause sequestrations of some proteins. It can also cause this can even cause a snowball effect. So there is not a direct contact like you see over here, but this is also getting stalled because of some secondary effect. So such things happen. So one of those ways was to first, so I try to take you through the genesis of this story so that we finally understand what is reticulo-mitophagy. One of the ways we started was to understand how the prion-mediated neurodegeneration occurs. Now, the prion protein, which is a cell surface GPI-anchored protein present in every cell of your body, of my body, of your pet dog's body, of the guinea pig that you might have in your house, every your cell has this GPI anchored cell surface protein. But perturbations in this protein, as we have known, in the form of the scrapie form of the prion, or by the presence of many mutations, can cause the formation of cytosolic prion. These prion proteins are detrimental for the cells, and they cause extensive neuronal death and neurodegeneration, which gets manifested in cattle as well as humans. You might have heard the name Kuru in a New Zealand tribe. Now, so what happens is that, so if we generate some transgenic mice which harbor these mutations in the prion protein, it simulates a very similar physiological, pathological outcome like the spongiform neurodegeneration that is present in prion-infected brains. These transgenic mice are also very short-lived, and they have all classical features of prion disease. So what happens in this disease is that what is seen is that if cytosolically exposed prion protein is present in cell, it causes extensive problems in cellular organelles. It stalls lysosomal degradation. So what do I see over here? On the top panel, you can see that the membrane, this is a normal cell membrane, which is marked by a green fluorescent protein by the fluorescently labeled EGF So the EGF gets uptaken by the EGF receptor, which is normally present on the cell. If everything goes on fine, in around 120 to 180 minutes, this entire green labeled EGF gets taken up and degraded at the lysosome. So by about 300 minutes, you see that this green color is almost totally drawn by the lysosomal degradation. However, if there is a faulty prion protein present, then this lysosomal degradation gets. So over here, we see in this bottom panel is that there is abundance of these EGF, green EGF inside the cell, which was uptaken by the EGF receptor but could not get degraded. So lysosomal degradation gets stalled. Not just that, this presence of this cytosolic prion protein also causes problems at the endoplasmic reticulum. The endoplasmic reticulum looks like this meshwork, like I had shown you in one of my first slides. However, if this cytosolically exposed prion protein is present, then it gets completely expanded, and this also is seen with many of the endoplasmic reticulum stress markers like the CHOP and the BIP proteins, which are classical UPR or unfolded protein response related proteins, which get upregulated when there is ER stress. So we find that there is a problem at the lysosomal degradation, there is a problem at the ER, and now finally I show you that there is a problem at the mitochondria also. So this is how mitochondria look like in a normal cell. However, if cytosolically exposed prion protein is present, mitochondria get perinuclear clustered. So, and it comes as like a cap on the nucleus. So there is problem at there is problem with lysosomal degradation. There is a ER expansion and ER stress, and there is perinuclear clustering. So why should these happen? To address that, what we found was that the cytosolically exposed prion protein actually stalls or sequesters the functioning of an a cytosolic E3 ligase. So this is an E3 ubiquitin ligase, which is responsible for ubiquitination of multiple cytosolic proteins and their normal functioning. However, when this prion protein is present, this ligase is kind of non-functional. And this also affects so if this ligase becomes non-functional, it actually affects the so I told you that one effect can cause a snowballing of multiple secondary effects. So if this ligase becomes non-functional, it actually stalls degradation of another E3 ligase, which is an ERE3 ligase. So I'll take you step by step through this. So first, let's see what happens when cytosolic prion protein is present. When cytosolic prion protein is present, so when prion protein is cytosolically exposed, it causes sequestration of this of this cytosolic ligase, and thereby it interacts with and causes problems in the cell. So how does it then affect the endoplasmic reticulum or the mitochondria or the so how does it affect? Now the question was to see the effect on the endoplasmic reticulum as well as the mitochondria. So let's take a look into these. So the cytosolic ligase actually causes ubiquitination and degradation of this ligase, which is GP78. This is an ERE3 ligase. So this ligase is present at the endoplasmic reticulum. This should have been proteasome. I'm sorry for it. This is a small mistake that happened. Anyway, so this ligase gets degraded via the proteasome, and the interaction between these two ligases and the degradation at the proteasome is all calcium dependent. So I'll actually jump a little and take you through and introduce you to one concept where we know that this is how the endoplasmic reticulum looks like. This is how the mitochondria look like. So the endoplasmic reticulum and the mitochondria are in constant contact with each other at various times. If these contacts increase, then it can lead to mitophagy. So what does the ER-E3 ligase do? The GP78 or the ubiquitin, E3 ubiquitin ligase, it actually leads to perinuclear clustering of mitochondria and mitochondrial fission. So mitochondria are going through constant fission and fusion cycles. Fission means they break up. So one mitochondrion will break up into two, and fusion is when two mitochondria combine together to form one mitochondria. So this process keeps going on. But if there is abundance of this ER-E3 ligase, the mitochondria can get affected. And it also has been shown to be responsible for the degradation of certain mitochondrial membrane proteins. And it can induce mitophagy. So now you see that abundance of an ERAD-associated ER-E3 ligase can actually induce mitophagy. So how does it all fit into the picture? So what is this ER-E3 ligase? GP78, or also known as the AMFR, autoprime motility factor receptor, is actually a transmembrane E3 ligase. It is present at the endoplasmic reticulum membrane. It is also present at the plasma membrane. It is linked to ERAD. It is also seen in certain tumor metastases. Now, what we find is that if the ligase, the cytosolic ligase that I just talked about, is depleted, or there is abundance of this ER-E3 ligase, then it gives the same phenotype. So the decrease in one or overexpression another, both have very similar phenotypes. Both give perinuclear clustering. So I showed you that when the cytosolically exposed prion protein is present, there is perinuclear clustering, and that we found was due to depletion of this ER-E3 ligase. But at the same time, what we found is that this depletion is also associated somehow with the overexpression of the ER-E3 ligase. So what we find is that if there is the cytosolic ligase is properly present, then the mitochondria are well distributed. However, the entire mitochondrial volume in the cell completely shrinks if there is a problem with the cytosolic ligase, or even if there is an abundance of the ER-E3 ligase. So here in these two cases, there is a problem in the mitochondrial distribution. However, it is fine when there is a mutation or an inactive form of this E3 ligase present. So what we found is that the cytosolic ligase can polyubiquitinate the ER ligase. It can interact with, as over here you can see there is an interaction between these two ligases, as is evident in the immunoprecipitation blot. And if we look for polyubiquitination, we find that the ER-E3 ligase gets polyubiquitinated by the cytosolic ligase. So, and naturally, when there is depletion of the cytosolic ligase either by a knockdown or by a knockout, or there are mutants which are inactive, ligase inactive mutants present, then the E3 present at the endoplasmic reticulum or G278 levels are very high, which means that there is a negative correlation between these two. And that correlation actually comes from calcium dependence of the cells. So mitochondria and ER, I have just shortly before told you that ER and mitochondria can crosstalk with each other. They form contacts with each other. These are the sites, these contacts are the sites where calcium buffering happens. If this calcium buffering, so if there is too much of calcium in the endoplasmic reticulum, that can get buffered into the mitochondria and vice versa. However, that can happen only to a certain extent, out of which, after which all hell will break loose and this calcium will leak out into the cytosol, and that will actually cause an elevation of the cytosolic calcium, which is extremely detrimental for the health of the cells. Thus, the cells will eventually go through apoptosis if too much of cytosolic calcium is present. So now here we use a drug which actually causes an increase in the cytosolic calcium. So this drug actually will release calcium from the mitochondrial stores. And so if we increase this drug, what we find is that the degradation profile of the ERE3 ligase decreases. And what we found is that however, that if we use a calcium chelator, then we can get back this. So here you can see, if we use a calcium chelator in the form of EGTA, which is cell permeable, you can actually get back the interaction. So if there is high calcium, no interaction, no degradation. However, if you use a chelator, then it comes back. So there is an opposite correlation, and this is all regulated by calcium. So what we showed is that under normal circumstances, when there is high amount of the cytosolic E3 ligase, a functional cytosolic E3 ligase, and a low ERE3 ligase GP78, then the mitochondria are healthy. As you can see, it is well distributed, and the mitophagy events are also very less. How do we see the mitophagy events? Here, the green marks the mitophagosome, where LC3 is a marker for the autophagosome, and here the red is the mitochondrion. So the red, you see that there are very small, less number of these reds surrounded by the green. However, when there is very high amount of this ERE3 ligase present, or low amounts of the cytosolic E3 ligase present, then such events where the LC3 surrounding mitochondria has increased drastically. So the mitophagy events have increased. So here we show that high levels of this ERE3 ligase actually causes increase in mitophagy, and this is very much related to the presence of cytosolic calcium in the due to paucity of time. I do not show you the details of how we show that calcium actually can regulate these mitophagy events. So to summarize, what I have shown so far is that when there is high cytosolic calcium, and when there is no interaction between these two ligases, which causes an abundance abundance of this ligase, that actually leads to the formation of mitophagosomes, whereby you can see an upregulation of the mitophagy events. Now, what are some of the physiological consequences of high calcium and increased GB78 levels in cell? Naturally, as I showed you, there is high mitophagy. There is also very high ER mitochondria interactions. These can very well be seen in the next slide, where in a normal cell, the ER mitochondria interactions are present, yes, but if you have very high levels of this ERE3 ligase, then such interactions drastically increase in a cell. So what happens because of this drastic increase in the number of such interactions and presence of this high level of the ERE3 ligase? What we find is that the presence of this high ERE3 ligase leads to unstable mitochondria. What do I mean by unstable mitochondria? Mitochondria become wobbly. Means, as you all know, mitochondria has an outer membrane. There is an inner membrane inside which there is the matrix or the mitochondrial matrix. Now, if this ERE3 ligase is present in abundance, then the outer membrane of the mitochondria become very unstable. This gives rise to escape of the inner mitochondrial material, which comprises the inner mitochondrial membrane along with matrix components and empty shells of these of the outer membrane. This can very well be seen in this leftmost panel, where if you look at the control cells, you can very well see that the red surrounds completely the green. The green is the inside of the mitochondria. The red is the outer part of the mitochondria. However, when there is high GP78 or very high levels of GP78, then what you have is that you can see that this arrow actually points the naked inner mitochondrial membrane, which we have termed as mitoplasts. So mitoplasts were people used to generate mitoplasts in vitro to do many physiological experiments, but this is the first time in the literature where under some physiological stress we have been able to show that mitoplasts can be generated ex vivo or inside culture cells. So here you can see that the inner mitochondrial material, which has completely escaped out from these posts. So these reds are, there are these portions of tiny red portions. Here in the black and white image, you can very well see this is a red ghost. The arrowhead actually points to the red ghost, and these greens are the escaped material. So under electron micrography, what we find is that in control cells, you can see ER mitochondria contacts, but when there is high GP78, you find fragments of the outer membrane. You find that the mitochondrion here is unstable. The outer membrane kind of separates out. So the arrowhead actually points an outer membrane kind of separating out from the rest of the mitochondrion. Over here you can see a full phago4 actually engulfing mitochondria. So these are actually destabilized mitochondria which are engulfed by an phago4. This will become clear shortly. Now, if we look, if we monitor these events over time, here the green marks the endoplasmic reticulum and the red marks the mitochondrion. Over time, what you find is that in the control cells, the ER and the mitochondria, they definitely come in contact like over here, but that contact breaches. So they again lose contact over time. However, when there is high GP78 or very high GP78, you can finally see that the endoplasmic reticulum in green engulfs the mitochondrion in red. So ER actually forms a phagophore around the mitochondrion. So you can see a reticulo-phagophore forming over here. So I have already told you is so the reticular part is kind of clear, part of it is kind of clear over here. So to further elaborate on this, we did not just by electron micrographs, we also studied by super resolution microscopy by structured illumination microscopy. Here the blue marks the inner mitochondrium, then comes the red, which is the, so here if you see, this is a 3D model rendition of the images that we generate. Here the green, the blue is the inner membrane, inner mitochondrion, the red is the outer mitochondrion, and the green is the endoplasmic reticulum. So in the control, what you see is that the endoplasmic reticulum is in close contact with the outer membrane of the mitochondria. However, in conditions when there is very high levels of this ER3 ligase present, the mitochondria kind of become this thing. They lose the outer membrane, and what you actually see is that the endoplasmic reticulum comes in very close contact, the ER coming, the green coming very close contact with the red, which is very unlike that happens in the controls. Now, to make it further clear, we use STED microscopy, which is also another platform of super resolution microscopy. Over here, the endoplasmic reticulum, which is in the green, can be seen. Actually, here if you see, the green is marking a hollow around the red, and here we show that the green protein is actually a reticulon family protein, which is FAM134B. The reticulon family protein is actually coming in very close contact with the inner mitochondrial protein, which is OPA1. So here the inner mitochondrion is coming in extreme closeness of the endoplasmic reticulum. So what we think is happening is that under normal situations, when the GP78 levels is low, the reticulon family protein is present and GP78 present in normal cell. Calcium is stored inside the endoplasmic reticulum or inside mitochondria. The outer membrane is proper, the inner membrane is where it should be. However, when there is stress, then calcium kind of escapes from mitochondria, from the endoplasmic reticulum. This actually causes elevation in the levels of the ER3 ligase, by which the mitochondrial membrane proteins are proteasomally degraded. Then the mitochondria become unstable or wobbly. The inner mitochondrial membrane gets exposed. Then it can actually come in contact with the reticulon family protein, FAM134B. So how does FAM134B help in reticulophagy? A very elegant paper came out. This is just a gist of it, where it was shown that this reticulon family protein, FAM134B, can actually associate with LC3 and help in the formation of an autophagosome. And this actually causes degradation of the endoplasmic endoplasmic reticulum itself. Why do you need degradation of the endoplasmic reticulum? When there is ER stress and there is ER expansion, then the endoplasmic reticulum needs to get a good portion of it needs to get degraded so that it can go back to its original shape and size that is usually present in a normal cell. So what happens is that actually what happens here in this case is that when a cell is stressed, then this reticulo-phagy protein, which is FAM134B, it recruits LC3 around the unstable mitochondria which have exposed inner mitochondrial membrane and form these phagophores, which are now actually reticulo-phagophores formed around mitochondria. So there is now a dual degradation happening, part of the endoplasmic reticulum getting degraded and unstable mitochondria also getting degraded simultaneously. So what you have is that here I show you that a part of the endoplasmic reticulum getting degraded along with these unstable mitochondria. At the same time, what we also see is that since GP78, elevation of GP78 was responsible for formation of these unstable mitochondria, the cell also causes a degradation of the GP78 rich endoplasmic reticulum, thereby again trying to downregulate the levels of this ER-E3 ligase so that normal situation can be brought back into the cell. We see this, we see that there is abundance of GP78 and inner mitochondrial proteins in lysosomal fractions of stressed cells. The outer mitochondrial membrane protein, which is marked by the, sorry, I would like to bring your attention to the top panel. Sorry, this is the bottom is the normal panel. The top is the panel where the GP78 levels are high. So here my MFN is a mitofusin 1 or an outer membrane protein. This does not get enriched in the lysosomal fractions, but GP78 also the inner mitochondrial membrane protein get enriched in lysosomal fraction, and this is also seen by lysosomal proteomic study where we have seen that if the FAM134B is somehow depleted by a knockdown, then the degradation of the mitochondrial proteins is reduced, and some of the ER proteins also reduces, but not much, only a few, but there is substantial decrease in the mitochondrial proteins. So when we actually categorize what type of proteins we see, we find that the outer mitochondrial proteins or the OMM proteins, which is in the dark gray, they don't get affected as much. However, there is a drastic decrease, a significant decrease in the inner mitochondrial membrane proteins as well as the matrix proteins. So, to summarize, what we find is that when there is abundance of cytosolic calcium, there is high levels of this ERE3 ligase. This ERE3 ligase actually degrades many of these the outer membrane proteins like the mitofusins by proteasomal degradation. That's why we did not see these mitofusins in the lysosomal fractions. So if we were to see in the proteasomal degradation pathway, we would see abundance of these mitofusins. So the ERE3 ligase actually causes degradation of the outer mitochondrial membrane proteins that exposes the inner mitochondrial membrane proteins. The inner mitochondrial membrane proteins can now come in contact with the reticulon family protein or FAM134B. Specifically, OPA1 forms contacts with the FAM134B. This is a part of the ER which starts forming the phagophore, then it completely engulfs the unstable mitochondria. Along with that, it also pinches off a part of the endoplasmic reticulum. So now what you have is, you have a part of the endoplasmic reticulum or the reticulo getting degraded, and mitochondrion, unstable mitochondrion or mito, also getting eaten up by this autophagosomal pathway. So ultimately, you end up having this dual regulation of reticulo-mitophagy. So this is the key player who actually identified this pathway of reticulo-mitophagy. Now coming to the most important slide, and that is the acknowledgement slide. All this work would not have been possible without the presence of these extremely dedicated students who are now very well settled in various capacities all across the world. So I have shown you the work of this student and this student primarily, Rukmini's and Devdatto's work, but I have just about touched upon Priyanka's and Xenia's work, where Priyanka actually did a beautiful job in showing how autophagosomes actually fuse with lysosomes. She has done a beautiful piece of work over that, and Xenia did a very elegant study, did a couple of elegant studies where she has shown how the expansion is dealt with in cells. I have not at all shown you the work of another very dedicated student, Devika, who actually dealt with something which is not all part of the story, where she showed how cell polarity is maintained, how actually that maintains the overall body axis symmetry, the left-right symmetric mechanisms. So she showed in ex vivo studies, but now after that, taking cue from her studies, there have been many studies across the world which have shown various organismal systems also that this can be recapitulated. These are the other people. So these are all people who have now left my lab, the alumni. Now these are the people whose work I have not touched upon. Rasdeep is actually working on mitochondrial regulation, the fission-fusion balance. Shiva Prato works on mitochondrial protein quality control. Palamo works on the mitochondria. So now you can see that the lab focus has shifted a little bit. We also work on mitochondrial regulate on the effect of mitochondria on cellular stemness. So which is actually being spearheaded by Dr. Devnita Chatterjee and also taken up by Nilanjan Das. I also would like to thank fantastic collaborators that I have had throughout. A good amount of analysis, 3D modeling, renditions, then molecular docking, protein-protein interaction studies, in silico modeling. All these are done in the laboratory of Dr. Shweta Chakraborty at CSI and IICB. The electron micrographs were generated by Dr. Munindra Bera. Dr. Shantanu Chandrajana helped us with the STED studies, and Rukmini, who was actually a PhD student with me, now collaborates with me on various capacities as in various projects as a postdoctoral fellow. So, and I thank my funding agencies, and these are few of the publications that have come out from the laboratory over the last 7 years or so. Thank you very much. I would be happy to take questions.
Thank you, ma'am, for your wonderful and enriching lecture. We have few questions from our audience. So with your permission, can we go through them?
Sure.
Okay, thank you. So, Akashtina Marik is asking, apart from neurodegenerative disease and cancer, do GP78 also play any role in infectious viral infection considering today's scenario? This question is on your
Uh,
screen.
I understand that. Not that I do not have an answer to that offhand, but yes, we are see actually partially knows what deals with something like that. So I will not be able to elaborate on it. So that is something which I do not think there is too many evidence in the literature.
Okay,
Okay.
thank you,
So
ma'am.
does that answer your
Uh,
question? Yeah.
so we have another question from Shahili Vishwas. She's asking, is there any mitochondrial protein degradation or how this autophagy gets affected in case of early apoptotic cells?
Can you just repeat the question? I did not understand the question completely.
Is there any mitochondrial protein degradation or how this autophagy gets affected in case of early apoptotic cells? Okay, I guess. Okay, due to some network issue, ma'am got disconnected. So to our viewers, please stay tuned. We'll be back soon.
Yeah.
So ma'am came back. I think there
Am
is
I
some
audible now?
network issue. Yes, ma'am.
Yes. Yes. Am I audible now?
Yes, yes, ma'am. Yes, ma'am. Audible.
Okay. So yes, mitochondrial protein degradation is definitely happens in the cell. Mitochondrial proteins get degraded piecemeal. Like I told you that there are various ligases which can actually get faulty mitochondrial proteins, or outer mitochondrial proteins, and kind of plug them out of the mitochondria and degrade them separately by the proteasomal pathway. Mitochondrial proteins can come out from the inner mitochondrial matrix or from the intermitochondrial membrane intermembrane space, or the IMS, which is the space between two the outer membrane and the inner membrane. So there are very well laid out mitochondrial protein quality control pathways. So they are not well understood yet, but they are definitely present. So inner mitochondrial membrane proteins can also come out from the mitochondria for degradation. Now outer mitochondrial membrane proteins can get degraded. Mitochondria as a whole can get degraded by mitophagy. So the protein degradation can be at various levels. Either the entire organelle gets degraded, or parts of the organelle can get degraded. Now, how does it affect early apoptotic cells? In the early apoptotic cells, mitophagy would be low, okay, or mitochondrial protein degradation pathway would be very well regulated. However, in during proper when full-fledged apoptosis is happening, then mitophagy events are also usually very high, because you have to appreciate that a lot of calcium actually leaks out from many of these autophagic cells, from many of these apoptotic cells. So that actually destabilizes mitochondria. And if that happens, then finally apoptosis will occur.
Oh, thank you, ma'am. So we have another question from Rahul Kumar. He's asking, what fraction of the mitophagy caused by the external stress comprises of MDB mediated process?
Uh, MDV process means?
Okay, he have another
Can
so
Rahul just elaborate
now
on that? I think I'm missing out
yes,
things.
he's elaborating in the next part.
Oh, okay,
He's
okay.
saying
Yeah, yeah,
in
yeah, yeah, yeah. Yeah. Sure. Yeah. Now I get back. Yes, Rahul. Okay. Uh, so reaction of mitophagy is caused by external stress. Like Ross, but what I can say is that external stress. External stress would eventually also generate internal mitochondria, internal stress inside, and that would go to mitophagy. But offhand, I do not know the answer to exact what percentage of external stress would lead to this mitophagy. No, no.
okay, ma'am. Uh, so we have the last questions from Prabhat Kumar Swain. He is asking, is reticulo-mitophagy is better option for dual organellar turnover mechanism in compared to other mechanisms?
No, this is not a better option. It is just one of the options. The cell can have multiple ways. This is one of the, I would say in this case, yes, slightly because you are down-regulating the endoplasmic reticulum problem as well as the faulty mitochondrial problem simultaneously. So this would be a better option in this case, but I do not say that this is the only thing going on. There would be various other ways also, others also happening. But yes, this is one of the studies to show that two organelles are regulated. So far, people knew about mitophagy. So far, people knew about reticulophagy. But here is a situation where the endoplasmic reticulum is stressed, the mitochondria are stressed. So why employ two different pathways? Why use up so much of energy to ultimately try to get back the situation to normal for the mitochondria as well as mitochondria? So if you have the two processes coupled into one, then you can simultaneously take down both these faulty organelles at the same time, and lead to and try to help the cell reach its normal equilibrium again.
Okay, ma'am. So these are the all the questions we have. Thank you, ma'am, for answering all these questions. It was great having you here.
Thank
I'm
you.
sorry that Professor NRA Banerjee could not join us today. She has passed on her good
No,
wishes to
that's
you.
fine.
Uh,
Yeah, that's fine. Thank you very much. I know that she has had issues. Yes.
so with that, now we will conclude this session. And thank you again, ma'am.
Thank you very much.
So for our
Thank you.
viewers, thank you
Thank.
for our viewers, thank you for joining us today. Join us 12th and 13th of June for the upcoming lectures of Ant Biology webinar series. Please like, share our Facebook page and subscribe to our YouTube channel and check our website www.zoologyhub.org. Our talks will be archived in the YouTube channel so that you can check them out later. And thank you again for joining us. Take care. Bye.